All right, well, let's go ahead and get started. I should be completely caught up on grading. So you should have everything back that you've turned in thus far. When I was grading though, I realized we still have 4 points unaccounted for in like the miscellaneous point section of the syllabus. So I came up with an assignment that hopefully doesn't feel like too much like busy work and helps you study for the final. So what I'm asking you to do is develop 4 questions that you think you might see on the final exam. Right? I'll have you develop 4 questions, any format you want. It'll be on the new material for Unit 4. You don't even have to submit the answers, right? I just want the questions. It's actually due on a Tuesday next week. That way by Wednesday, I can compile all of those, post it onto Moodle, and you basically have like an extra little study guide. Questions made by your peers. And it's possible if I love those questions, I may pick up all of them on the exam. So that's a great way to study, and it should be an easy 4 points. Like I said, you don't even have to provide the answers. You just have to come up with 4 questions. All right. That'll be the last miscellaneous points that I had to dole out to you all. So otherwise, all the points should be accounted for in the assignments you've either done or will be doing in the last half of the semester, which is really just scientific literacy and 2 other content checks. Any questions about this assignment, other assignments? All right. So with this class here, my other class is a little ahead of you guys, so we're going to jump back and talk about the last bit of natural selection, which is sexual selection, right? It's a form of natural selection, and this is where we would talk about individuals that have certain traits are more likely to engage in successful reproduction than individuals that don't have those traits. So in many species, right, this is very common in the animal kingdom. In many species, this affects the male characteristics more than the females. Right? So if we look at a couple of examples, we might have intrasexual selection, which is male-to-male competition. So an example of that might be these crabs. They have these really large claws. The larger claws help them to fight each other when they're fighting for potential mates. Right? Same with like deer and antlers. Right? Antlers are only on the males. It helps to fight for potential mates. This is different than intersexual selection, which is where it's kind of the female's choice. So the males typically put on a very big, elaborate display, like this peacock here, right? The male peacock is the one that has the elaborate display of feathers, and then the females choose based on the display of feathers, or based on like a mating dance. I don't know if you've ever seen videos of like jumping spiders trying to mate, and they do these very elaborate dances with their arms and their abdomens, right? But it's typically always the male doing this, and then the females choosing who they want to reproduce with. Yeah?
What do turtles do?
I have no idea what turtles do. That's a great question. That might be a question for Google, because I do not know. All right. So this would be a form of non-random mating, right? Meaning that who you're mating with is not random. There's a preference in who you're mating with. Now there's 2 different patterns that can emerge with non-random mating. Individuals can mate assortively, which is where individuals with similar phenotypes will mate with one another. So an example of beetles here, right? These beetles, they have the same coloration, right? They mate with one another. They have the same phenotype. This is a good example of assortive mating. Right? And the opposite end of that, maybe certain species, like individuals with dissimilar phenotypes. Right? This would be an example of disassortive mating. Right? So in this case, I have 2 beetles here. One's brown, one's a lighter brown, different phenotypes. Right? That difference is attracting them, and thus they are going to mate and create their offspring. So it can be assortive, it can be disassortive, based on whether or not your phenotypes are similar or dissimilar. Now another type of non-random mating is inbreeding. This is where we are mating two genetically related individuals. So if we look at an example of inbreeding, I feel like I'm going pretty fast, so I'm going to just pause a little bit here. So assortative versus disassortative versus inbreeding, right? What inbreeding tends to do in populations of species is that we're not really favoring a particular phenotype, but rather we're losing the heterozyte genotype, right? So if we say look at a population here, now this is a population of plants. They're able to self-fertilize, which means they're fertilizing themselves to produce offspring, so very genetically similar. We might start off with a generation that has more or less an even spread of homozygous, recessive, and dominant to heterozygous individuals, right? We see it's pretty evenly spread out. But over successive generations of inbreeding, we end up with a population that has very few individuals that are heterozygous. Instead, we're only homozygous dominant or only homozygous recessive, right? So we tend to lose individuals, we tend to lose that heterozygous genotype over time. Now you might be wondering, why is that a problem, right? We still have both phenotypes in the population. Well, it's something to do with a concept known as heterozygote advantage, right? Where when you have a diversity of alleles in your genotype, so you're not homozygous, but rather you're heterozygous, you tend to have more alleles that are beneficial at disease resistance, right? So often we get these loss of function mutations, right? They might show up when you only have the dominant allele, or when you only have the recessive allele. Very commonly, it's when you only have the recessive alleles. An example of this is the panthers, right? There are a population of panthers in Florida. Their population was pretty small. So small, in fact, that they had to inbreed with each other. There were no other possibilities for mates. And what we saw is the survivorship of the offspring over the years really dramatically decreased, right? So something about their genetics when they were inbred, right? These diseases were not beneficial at all. In fact, they led to the decline of this population. Now, what researchers did was they actually took panthers from a population in Texas, introduced them to this population in Florida. So now we're increasing the diversity of alleles, and we see that that population rebounded a bit, right? We had much higher survivorship of these panthers because we increased that genetic diversity, right? So inbreeding can have these negative effects, especially if there are diseases associated with particularly the recessive allele. All right, this is different from random mating. Random mating is where individuals are likely to mate with any other individual in a population. There's no preference shown, right? In nature, this is very rare. We don't often see this within particular species. Really, we only see it in species that don't have eyes, for example, or do a type of spawning. So they, especially in aquatic species, they just release their gametes into the water, hopes that an egg and a sperm will connect and make a zygote and another individual. They have no idea who they're mating with, right? So this would be an example of random mating. Again, this is very rare. This does not happen for the majority of species that we know of. All right, so non-random mating versus random mating. It's non-random, it can be disassortative, it can be assortative, we can have inbreeding, which can lead to genetic problems. random mating, we're equally likely to mate with any other individual in the population. All right, let's apply these concepts. I want you to answer a couple of questions that will apply these concepts to a couple of different situations.
Similar ones is the and then I'd be random because they don't have eyes and they just go into the water. So
Can't figure out if it's here or maybe drifting in from the scenery.
So there's coral polyps, which is when coral reproduces. This is the life form they take on. It looks very similar to an anemone.
All right, I have two different pairs of frogs. One pairing is a green frog with a green frog. The other pairing is a green frog with a black frog. Which is disassortative mating? The left one, which is what two colors? I gave you the answer. Two colors, right? Two different phenotypes, right? That would be disassortative mating. All right, these coral polyps, so when a coral reproduces, they produce these polyps. They release their gametes into the water, right? Is this an example of random or non-random mating? Random, yeah. They don't know who they're mating with. They don't even have eyes, right? They have no idea who they're mating with. All right, any questions about sexual selection? If you're on the biology track and you take ecology, evolution, and behavior, you'll dive deep into this because there's some really interesting strategies and patterns that emerge in different populations. All right, so that was the last bit of natural selection. So we're going to move on to another mechanism of evolution. Right? We're going to start with talking about genetic drift. Right? So we've talked about natural selection. We're moving on to genetic drift. And genetic drift is where we have a change in allele frequency. So evolution is occurring, but now it's due to random chance. Right? The random events, that they're completely random, right? There's nothing suggesting that one phenotype is preferred over another phenotype. All right, so if we're looking at a population of rabbits, right, we don't care if the rabbit's fur color is brown or white, right? Some event has happened, completely random, irregardless of what the rabbits look like, that lead to changes in allele frequencies. Now, this typically happens in smaller populations. So here I have a graph showing three different populations, right? The two populations on the red lines only have a sample size of 10, versus that population in the middle has a sample size of 1,000, right? And specifically, we're looking at examples of different mice populations. In the smaller populations, over many generations, so this is over 50 generations here, we see that in the small populations, we're either going to tend to have populations that are either all brown or all white. We've lost an allele and we've fixed the other, right? So in the white population, white is recessive, indicated by the lowercase b's, right? We have completely lost the dominant allele, and we have fixed the recessive allele in place. Same is true for the top population. We only have the dominant allele. That one is fixed in place. We have lost the recessive allele. All right, so those populations, they started out with 10 individuals. But if we look at that population that started out with 1,000, we see the proportion of dominant to recessive alleles is pretty steady, right? There's really not a change. there are more individuals in this population. It adds kind of a protection buffer in the allele frequencies. Right? We do not see a loss or fixation of an allele. So genetic drift, these random events, whatever random event has caused a population to be all brown or all white, right, is going to show up in small populations. Larger populations really not going to be affected, and if they are, it's going to take a lot longer. All right, so what do I mean by a random event? Right? Well, a random event might be that you have a population of beetles and you take a step and you don't know who you're stepping on, but all of a sudden you feel that you're crushing some beetles. And it just so happened that the beetles that you squashed were green, which means the population left after you squashed some of those beetles are mostly brown with fewer green individuals. That is completely random, right? You weren't looking where you were going. You weren't trying to squish the green beetles. It just happened that that's where you stepped. Right, so genetic drift is altering that genetic frequency. And we can look at two different effects. Right, so genetic drift can occur as a bottleneck effect or as a founder effect. Right, this example on the screen is an example of a bottleneck effect. And what is a bottleneck effect? So a bottleneck effect is when a population is dramatically reduced in size, right? So in this example here, I have frogs in a pond, right? In my top pond here, I have 3 different phenotypes. Looks like they're green, they're yellow, or they're this brown striped color, right? So that's my starting population. I have 3 different phenotypes. Now, during the summer, it didn't rain. There's a little bit of a drought. The pond has shrunk in size. Right? Now, the amount of rain that did not fall on the environment didn't care about which color the frogs were. Right? The rain does not care if the frog was green or yellow or brown. But what had happened is that that pond shrunk, and it just so happens, completely random, that all of the green frogs died off. Right? So now in the fall, maybe we get a lot of rainfall, that drought ends, the pond fills back up, that population can rebound, right? And now we see that the population only has yellow and brown frogs, right? It doesn't have any green frogs because they were killed off. So we're lowering the genetic diversity here, right? So we start off with a population with a lot of genetic diversity, something random happens, right? It's a drought, the rain kills off half the frogs, more than half the frogs. All the frogs with the green allele are dead, they're gone. So now that population, when it can rebound, does not have that green allele. All right, so surviving members may have different allele frequencies from that original population. Now this is different than a founder effect. Right? A founder effect is not where individuals die off, but rather a subset of individuals from a larger population leaves and forms a new colony. Right? So if we look at this population of butterflies up here, all right, we have these butterflies. They have alleles for butterfly colors of orange, black, or white. Now a subset of these individuals, right, flew to a different island, right? Maybe they went out exploring and found a better habitat. Or maybe they got caught in a gust of wind that blew them off course, blew them to a new island, right? It was random in what phenotypes founded that new colony, right? And it just so happens that that new colony was founded without any butterflies with the white phenotype, right? So that population, when it rebounds and adds more individuals to its butterfly population, is just not going to have the phenotype for a white butterfly, right? So it has less genetic variation than the original. Now, an example of this in humans is actually in an Amish population, I believe in Pennsylvania, right? The founding families that founded this original population in this geographic area had the allele for having more than 5 fingers on your hand, right? Now this is a recessive allele, right? It only shows up if you have both recessive alleles. And it just so happens that these individuals that founded this establishment, right, this population of Amish individuals, had that recessive allele phenotype, right? And so that Amish community is pretty isolated. They were mostly stick to themselves, right? As the population grew, we saw an increase in the number of individuals that had more than 5 fingers, right? So this is an example of a subset of a community, subset of the Amish population, founding a new colony, founding a new population, and it just so happened that that population had that recessive phenotype show up. All right, so I realize I didn't put a timer on this, but I'll do like 2-3 minutes here. I've given you a couple of different scenarios. I want you to tell me, is this genetic drift, right? And if it is this a bottleneck or is it a founder effect? And these types of scenarios are the types of scenarios that I could present to you on an exam, right? Describe a population and ask you what is going on here.
It's not.
Yeah. Well, we'll be recorded. Back out. It's up to you.
Yeah, it wouldn't be random effect.
So, what was it called when the wolves specifically went after the
I mean,
yeah. Was it specific?
it could be genetic drift, but it wouldn't be the founder effect or the bottleneck effect. It would just be random. Genetic drift.
So the first one's not.
Yeah, what is it? An example?
Is it natural selection? I thought there was like a more specific.
The environment, predators.
Can it be genetic drift without the bottleneck after the founder effect?
Yeah.
Good question. I don't want to say outright no,
Because
but I think it'd be very
I
rare.
feel like the example, like the first example, it could be genetic drift, but it wouldn't be an example of bottleneck effect.
I would know.
Because the population is being decreased.
It is being decreased, but is that decrease random? Targeted. Yeah, the environment, or specifically the predators, are doing the selecting here. Would be natural selection. Yeah, because it's not completely random.
Andrew still can't get out of his head that it killed me 3 times in a row. I did. You know what you said?
Bottleneck. They're separated.
Not that one
Because
dude.
the population reduces dramatically and because if you think going on the bottom, being the neck of the bottom, there's less to affect less.
All right, let's go over these scenarios here. So in that first scenario, we have a population of frogs, right? They are 2 different colors, and there are predators in this environment that prefer to eat the dark-skinned frog, which means over many generations, that population evolves to have mostly lighter-skinned frogs. Is this an example of genetic drift? No. Now, what is it an example of? Natural selection, yeah, it's the environment doing the selecting, specifically the predators in the environment doing the selecting. All right, the other example here is a real example. This actually happened. So northern elephant seals, their population was reduced to 20 individuals at the beginning or at the end of the 19th century. But that population has rebounded. There's now over 30,000 individuals. This is genetic drift, but is it a founder effect or a bottleneck effect? It's bottleneck. Some individuals died. That population reduced in size, right? It is a bottleneck effect. All right, any questions pertaining to these scenarios or genetic drift in general? All right, we have two other mechanisms of evolution to go over. The next one is gene flow. All right, so we've gone over natural selection. We've gone over genetic drift. We're going to talk about gene flow. Gene flow is almost kind of a fancy word for migration, right? It's the change in allele frequencies between two different populations when migration can occur, when mixing of the populations can occur. All right, so migration actually tends to reduce differences in allele frequencies. So in either population, it tends to increase genetic diversity. So if we have an example here, we have two populations of deer, right? One is this western deer population. Some environmental pressures have been acting upon it. It has been fixed, so they only have lighter color or mainly lighter colored deer. Right? The eastern population is the opposite. They mainly have darker colored deer. Right? Now, normally these populations are kept separate, which is why those two populations have different frequencies of light and dark fur or hair. I'm not sure what it is for a deer, right? But at some point, they are able to come into contact with each other, right? Maybe they are on this mountain range and all of a sudden there's a pass that makes it easy to traverse this mountain range. So now we can have migration of individuals leaving one population and entering the other. So what this does is in a population of predominantly darker deer or predominantly lighter skinned deer, right, we can now reintroduce the lighter deer or the darker deer allele into that population. Right? So both populations now have increased genetic diversity. Right? We can kind of see that represented as dots on this top here. Right? Population 1, some sort of geographic barrier. Population 2, when migration can happen. Right? Now some of these red dots can enter into the blue dot population and vice versa. All right, if we look at more examples of migration, this pie chart in the corner here is, I think it's basically showing you that deer population we looked at before. All right, so if we look at frequencies of alleles, one allele is represented by being blue, one allele is represented by being green, right? We see that two populations, they're different in the number of alleles they have for each type. But all of a sudden, they're able to migrate and mate with one another. And we see the resulting frequencies in both populations have been changed, right? We've increased our genetic diversity. Same if we look at this example with birds, right? So we have this blue bird here, population A. Some selection pressure has created this population to only have homozygous dominant individuals, right? Opposite is true for this red bird. Something has caused them to only have individuals that are homozygous recessive, right? Now, if mating is able to occur between these populations, right, somehow they're in close enough proximity, they can interact, they can migrate. The resulting offspring of a blue bird going into this red bird population, or the red bird going into the blue bird population, is offspring that have a heterozygous in their genotype, right? So they have a recessive and a dominant allele. We have introduced that allele back into the population. Again, increasing that genetic diversity. All right, so gene flow, we're talking about migration. between different populations, we're increasing that genetic diversity. All right, we're going to move right on into the last type of evolution, mechanism for evolution, and that is mutations. All right, we've already kind of talked about mutations. Right, so mutations is something that has occurred in our genetic code that can introduce a new phenotype, and it's completely random. Right? So again, if we're looking at an example, I have an example of mice up here. They are all brown mice, right? In their genetic code, maybe that codon that specifies for brown, that amino acid sequence, is GGG. Now all of a sudden there's a mutation, right? We've mutated one of these G's to become a C, and it just so happened that that correlates with fur color. That changed a brown mouse to become a white mouse, right? Now this is completely random, right? The mice did not come together and have a conference and say, we need to create a white mouse. That did not happen. This is just completely random by chance. But as a result, the successive generations now have that new phenotype. Right, now have white mice introduced into that brown mice population. Right, so it's completely random. It can be beneficial or it can be detrimental. Right? We talked about this with mutations. So if we think about what if this mutation happened in an environment that was predominantly darker in color, right? That mutation of being white is not going to be a good mutation. You're going to stick out like a sore thumb. Predators are going to be able to find you very easily. Versus the opposite, right? Maybe this population was in a lighter environment, and now we have this mutation for a white mouse. That's probably a beneficial mutation. They're going to find it easier to hide from predators than the original brown mouse. All right, so again, completely random and can be either beneficial or detrimental. Now, often if it's detrimental, that phenotype will likely die out after a few generations. It'll just be selected against via natural selection. All right, so we've talked about 4 different mechanisms. All right, I want you to tell me for each mechanism of evolution, is it increasing or decreasing the genetic diversity in a population over time?
Alright, this is what you saw. This is what you're telling me.
Is that the whole point of it? Is it either goes to 0 or 100? Yeah. So yeah, it does decrease it.
Listen, Andrew.
And then 12 gonna increase. Yeah.
So you sound like this.
So more similarities.
Yeah. It can be beneficial.
Yeah.
That's exactly what it sounds like.
Last night, got tiny hills on
All right, we have 4 different mechanisms of evolution. Natural selection, we'll do like a thumbs up if it's increasing variation or thumbs down if it's decreasing variation. So natural selection, increasing or decreasing? Yeah, for the most part, it's decreasing. right? But if we're thinking about different patterns, if it's a selection that maintains the allele frequency, it's that balancing pattern, right? Then we can actually maintain genetic diversity. But if it's directional, stabilizing, or diversifying, we are decreasing our diversity. What about genetic drift? Increasing or decreasing? Decreasing, yes. Gene flow. Increasing. We're bringing in more alleles to a population because of migration. And mutations? Increasing, yes. All right. Any question about these mechanisms of evolution? We are about to jump into a kind of a new topic. So we're going to take a closer look at population genetics. So we're going to take a closer look at the frequencies of having dominant and recessive alleles or different genotypes within a population. All right, so just to reiterate, what is a population? We talked about those Joshua trees here, right? It's a subset of individuals, one species, occupy the same geographic area, and have the ability to reproduce with one another. Right, now these have to be a group of sexually reproducing species. If they reproduce asexually, right, then you're really just creating a colony of individuals that are cloned, that have the same genetics, right? But a population would be a group of sexually reproducing individuals. They can interbreed, which means they're the same species. They have easy traveling distance. They have the ability to interact, which really relates to the biology of an organism, right? If we think about how far a mouse can travel versus a human, right? Humans, we can circumnavigate the globe at this point. Mice, it would be very hard for them to do that on their own. Same with like the coyote here. In the US, we tend to have 3 larger populations of coyotes. Actually, I think these are wolf populations. We typically have an East Coast one, a Southwest, and a Northwest population. All right. So really easy for them to have these large migration ranges, right? Their geographical range is huge, but it's hard for them to interact on one side of the country versus the other. So scale for different organisms can be very different. All right, so if we're looking at a population and we're specifically looking at their genetics, we have to look at their gene pool. So their gene pool is all of the alleles for every gene in a given population. Now, often we're not looking at every gene at once. We're often going to isolate one gene to look at a time, just for simplicity's sake, right? But a gene pool would be every allele present for every gene in a population, right? So by looking at a gene pool, we can look at the population genetics. We can look at the variation in the gene pool and how that variation can change from generation to generation. So if we look at this image here, right, the geographical boundary of this population of beetles is highlighted by this brown area here, right? That is our population. If we look at the gene pool, we're specifically looking at their genotypes and whether or not they're homozygous dominant, homozygous recessive, or heterozygous individuals, right? Or how many dominant alleles do we see versus recessive alleles? Right, so often we'll see emphasis on alleles rather than genotype, but we can calculate both. All right, so population genetics, we're looking at that gene pool. As a reminder, genes are typically what we call polymorphic, which means there is more than one allele. Right? At least 2 alleles, if not more, that lead to different phenotypes when matched together. So a polymorphic gene is 2 or more alleles, right? A gene that has 2 or more variations. So in this population of flowers up here, we can have flowers that are either red or they're yellow, right? They have 2 versions of their flower color allele. which is different from populations that are monomorphic, right? So these birds that I have in the image here, these Coriolis, they are monomorphic in their plumage, which means they can only be white. They do not have another allele for a different color of plumage, right? They are monomorphic in the plumage gene. Now this population could at some point get a mutation, often caused by a point mutation that we talked about last unit, and could develop into a polymorphic gene. But for now, there's only one version of the allele. All right, so polymorphic, more than one version, monomorphic, just a single version. We're going to focus on genotypes that are polymorphic, genes that are polymorphic. So how do we actually analyze genetic variation in a population at a given time? Well, we look at frequencies, right? We look at either the allele frequency, so how many dominant alleles are in the population, how many recessive alleles are in the population, or we can look at genotype frequency, right? So how many homozygous dominant individuals are there? How many heterozygous individuals? How many homozygous recessive individuals are there? Right? And we do this using math, all right? So we are going to calculate out some of these frequencies for different populations. Oh, I thought I had one more thing that popped up there. Apparently I don't. All right, so we have different equations that we can apply to populations to figure out what their allele frequencies are, what their genotype frequencies are. Right? So if we first start with allele frequencies, right, we would have an equation where we would look at the number of copies of a specific allele in a population. Right? So maybe we have a population and we're only looking for the dominant alleles versus the recessive alleles. Right? So we would say we're looking for the dominant alleles. Right? We would put the number of copies of that dominant allele in a population on top, and divide it by the total number of all the alleles for that gene in a population on the bottom. Right? If we divide that out, we would get a decimal number that we could convert to a percentage. We'll do some examples of this. You can do basically the same thing for genotype. Right? So genotype, now we're looking at individuals. Right? Are they homozygous? Are they heterozygous? So we would say count out the number of individuals that are homozygous dominant and put that on top and divide it by the total number of individuals within that population. And again, that would give us a decimal, which we can convert to a percentage. We could say 45% of individuals in a population are heterozygous for that specific gene. Right? So we have these fancy equations, but they're not too hard to apply to real-life situations. I know this slide has a lot of text. So allele, just looking at dominant versus recessive, or one type allele versus the other type of allele. Genotype, looking at the combination of alleles in individuals. All right, best way to learn these are to practice. So we are going to practice using a population of flowers. So these are 4 o'clock plants, and we're going to look at a population of 100 of these plants. Now, this population has conveniently already been genotyped for us. We know exactly how many individuals are red, how many individuals are white, and how many individuals are pink. So notice, when we have our genotypes, they're both capital letters. So capital R, or capital W. So why are they both represented as capital letters? Does anybody remember what this type of dominance is called? Not codominance. Incomplete dominance. It was on the same slide as codominance. Incomplete dominance. So when they're both dominant and they produce offspring that have one of each allele, we're mixing that allele together to produce a different phenotype, right? So a red plant or an individual that has red and a white allele together would make a pink flower. All right, so we know the genotypes of these individuals. We know that there are 49 of them that are red, so capital R, 42 of them that are pink, so capital R, capital W, and only 9 of them that are white, so capital W, capital W. Now, important thing to remember when we're calculating out allele frequencies is that homozygous individuals are going to have 2 copies of that same allele, right? So they're either going to have 2 dominant alleles or 2 recessive alleles. Versus a heterozygote is only going to have 1 copy of each, right? A dominant and a recessive. You'll see why this matters when we move on with our example in the next slide. All right, so we know how many individuals for each genotype we have. We want to calculate the allele frequencies. So what percentage of red alleles are in the population, or what percentage of white alleles are in the population? So we're going to go through an example, and we're going to try to figure out what the allele frequency is for the white allele. And we're going to apply our equation, right? So when we look at our equation, we first have to identify out which genotypes contain the white allele. Well, we know that there's 2 genotypes that can contain the white allele. You can either be homozygous, right, for the white allele, so have 2 white alleles, or you can be heterozygous and have 1 white allele. So if we were to plug this into an equation, right, we want the total number of alleles of just the white allele on top. So we would take the number of individuals that are heterozygous, right, they have an R, they have a W, plus 2 times the number of individuals that have the white allele. Now we're multiplying this by 2 because it has 2 of the white alleles. We don't have to multiply that first one because it only has 1 white allele. All right, we would add those together to get our number, and then we would divide it by the frequency of every single allele in the population. Now remember, each genotype has 2 different alleles, so we're going to multiply each genotype by 2 to capture all of the alleles. What we end up with, right, if we plug in all the numbers, do all the math, we get 60 divided by 200, which is 0.3 or 30%. Right? So we can just plug in the numbers and you'll have an opportunity to practice this. Genotype is done pretty much the same way. Right? We count out the number of individuals with a particular genotype. Now we don't have to multiply by 2 because we're only looking at the combination of 2 alleles together. So if we're looking at the frequency of white and white, right, having a homozygous phenotype for the white allele, we know that there are 9 individuals. We plug that in on top. We divide it by all of the individuals in the population, which 49, 42, and 9 added together is 100 individuals. We get 9 over 100, which is 0.09 or 9%. All right.
Why is for the total population on the bottom first for the frequency, why is it 200 not 100?
Yeah, because for genotype, we're looking at individual genotypes as a whole. But for the allele, we have to times it by 2 because each genotype has 2 alleles. Yeah. All right, so let's practice this. I have given you the example we walked through where we looked at the white allele. I want you to calculate the frequency of the red allele. I'll give you a few minutes to do so, and then we'll walk through it together.
Anyone divided by Shit, life. The full. Shine it. Wait. I cannot.
For which one?
So,
On the top? Because there's 2 of the white alleles we have to count. Versus that heterozygote only has 1, so we don't have to multiply it by 2.
What are you doing, bro? On the job. Why is he depressed? Why is he depressed?
Yeah, 0.3. All right, did anybody come up with an answer in that time?
70%.
70%, yeah, that's exactly right. Right, so it's 70%. So if we did the math, right, we would apply it to the equation that I've written up on the board. We look at the number of heterozygotes, right, which only have 1 of the red alleles. So that would be 42 individuals, plus 2 times the homozygous for the red allele, because there's 2 alleles we have to account for. So that would be 2 times 49, divided by 2 times all of the alleles, right? So 2 times, we have to count for both alleles in each genotype. 2 times 49, actually I have my white one as first, so 2 times 9, plus 2 times 42, plus 2 times 49. Right? So then if you did all the math and simplified it down, you would essentially get 140 over 200, which equals 0.7, times it by 100 is 70%. Now, did anybody find the answer in a different way?
No. You can assume since the other one's 30%, that's 70%, right?
Yeah, we have to count for all individuals in a population. If we know 30% of them have the white allele, we could simply say the other bit, or the other half, 70% half, has to be the other red allele. Much easier, and I purposely withheld that information from you to make you try doing it the hard way first. Right? So, any questions on how to calculate these? Again, best way to do this, just practice. All right, we are going to move on to talking about the Hardy-Weinberg equation, right? So the Hardy-Weinberg equilibrium is where we are now looking at a population, and we're trying to develop or trying to look at the allele frequencies and the genotypic frequencies in a population, but that population is not evolving. It's staying the same, right? There is no change in allele frequencies from generation to generation. Everything is staying constant, right? So again, developed after these two individuals who made this equilibrium hypothesis, developed out some mathematical equations that, yes, we're going to go through. Right, but the key here is the population is not evolving. So if a population is not evolving, that means there are no new mutations occurring. No selection is happening, right? The population tends to be large, which means genetic drift does not have an effect. There's no migration between different populations, so there's no gene flow, and all the mating is random, right? In reality, this really doesn't happen, right? Populations rarely reach equilibrium. It is hard for all of these conditions to be met, right? It is hard for a population to simply not evolve either by natural selection or random chance or migration or random mating, right? So if we say plug in some numbers here, look at an image, you can say looking at the initial allele frequencies in a population from our gametes, right, of dominant versus recessive would be 0.6 and 0.4. If we followed this population, right, gametes to zygotes to juveniles to adults, the final allele frequencies are going to be the same. They're going to be 0.6 and 0.4 respectively. All right, so in reality, if populations rarely reach equilibrium, why is this equation useful, right? Why bother calculating out equilibrium in a population? Well, if you're a population scientist, you're a population ecologist, and you are going through doing your calculations, and you find that your population, let's say this population up on screen here with the initial allele frequencies of 0.6 and 0.4, now have a frequency of 0.8 and 0.2. It has changed. What that means is that there is some sort of evolutionary mechanism at work. So if you are a population scientist studying a population and you notice that your population is not in equilibrium, your next question would be, what mechanism is at work, right? Did this population have a mutation? Was there migration between two populations? Was natural selection occurring on some phenotype? Right? It allows you to ask this question. What is happening to make it not in equilibrium? All right, so we're moving on with the assumption, again, that this population is not evolving if it's in equilibrium. We can apply some equations to a population, again, that's not evolving, it's in equilibrium, to calculate the allele frequencies and the genotype frequencies. And these are equations that I would expect you to know and be able to apply, which is why we're going to spend some time practicing. So if we were to look at the allele frequency, we could say P plus Q equals 1, right? We could say P represents one allele, the Q represents the other allele. So in an example, if we have a heterozygote with a capital A is the dominant allele, lowercase a is the recessive, we could assign P to the capital A. We could assign Q to the lowercase a. Oops, pressed the wrong button. Right? So P plus Q equals 1 then becomes capital A plus lowercase a equals 1. Same for the genotypic frequencies here. Right? That equation is P squared plus 2PQ plus Q squared equals 1. P squared indicates that we have two P alleles, so that would be, say, homozygous dominant individuals. 2PQ indicates we have one of each allele, so that would be the heterozygote. And then Q squared, because we know Q is assigned to the recessive allele, would be the homozygous recessive individuals. All right, again, best way to understand this is to put it into practice. So if we, again, look at our population of 4 o'clock plants, we're going to look at that exact same population. We already know the allele frequencies. We've calculated this out using the other equations. We know it's 0.7 and 0.3 for for red and white respectively. All we have to simply do is just plug in the numbers to our equation to calculate genotype frequencies. Right? We'll assume that the P is for the red allele and the Q is for the white allele. We just have to plug in the numbers. So P squared is the genotypic frequency of red and red. 2PQ is the heterozygote, so red and white. And Q squared is the homozygous individuals for white, right? White and white. So if P equals 0.7, P is the red allele, and Q equals 0.3, and Q is the white allele, we can plug it into the equation and say P squared is 0.7 squared, which equals 0.49, or 49% of the population has this genotype. Right? Same for the heterozygote, same for the homozygote for white. Now, because this population started out with 100 individuals, you'll notice that these numbers sound familiar because we use these numbers in the other equation, right? That was just by chance. We started with a population of 100 here. But we could use the same equation if the population started at 112 or 157, right? And essentially we would see that this is we could put this into a Punnett square and calculate out what the offspring could be in a population, again, that is not evolving. So I'm going to explain this figure on the next slide here. All right, we've done all our calculations. Those calculations, I put them at the top of this slide so you can again see them. All right. We could say that in 2 generations for 2 heterozygous individuals, right, so they have the red and the white allele respectively, we could calculate out the percentage of individuals, percentage of offspring, that will be red and red, so P squared, which would be 49, right? 21% for the heterozygous individuals, and we have 2 heterozygous individuals in our Punnett square, each at 21%. And then the probability of being homozygous for the white allele would be 9%. All right, I know there's a lot going on here, so we're going to continue with examples, right? This is something I might ask you to calculate on an exam, and yes, I am bringing calculators to the final exam for this very reason. But a good way to double check your answers is you can add up all the values and see if it equals 1. Right? And again, population is not evolving. We're going to start class on Friday going through an example step by step applying these equations, so you'll have an opportunity to learn them even better.